For this section of notes today, we are going to be covering the second half of protein synthesis, which is the step of translation. So if you have not already downloaded the Unit 3.2 guided notes that were begun on Monday, please go ahead and download those now from Google Classroom or pick up a paper copy at the front of the classroom. On Monday, we covered the first two learning objectives for RNA and protein synthesis. Today, we're going to look at the next two, so explaining how the genetic code works and describing the role of ribosomes in assembling proteins, which is the process of translation, or part two of the overall process of protein synthesis. So let's look at what translation is. Translation is the process of going from an RNA molecule to a protein. So we learned in the last section of notes that transcription is taking our DNA molecule and turning it into an mRNA strand. And this is the process of transcription. But today we're going to look at how the cell translates that RNA into a new language, a new molecule, which is the protein. And remember, proteins are the building blocks for our cells. They have hundreds of different functions inside of our bodies, and we cannot survive without those proteins that are made during the process of translation. So the definition for translation is, putting that together, the process where proteins are made from the RNA, and this takes place on the ribosomes. Ribosomes are a cell structure, a cell organelle, that can be found in the cytoplasm, or they can be attached to the endoplasmic reticulum, or we call the ER. A ribosome is actually made up of two parts. You can kind of see it in this picture here. We have the top part here, which is called the small subunit, and then we have the bottom piece here, which is the large subunit. And together they attach to the RNA strand that was created in the nucleus during transcription. As the ribosome reads this RNA strand, molecules of tRNA are going to come in and also attach to the large subunit of the ribosome and carry with them the amino acids that are needed to build the growing protein strand. We'll look at this in a little bit more detail. So a ribosome actually has a little bit of a reading disability. A ribosome is only able to read in groups of three. It can only read three-letter words. Imagine your whole life you could only read words that were 3 letters long. And that's really what a ribosome is. So it has to take that really long sequence of RNA, which is made up of, you saw in the activity on Pivot, it could be thousands of base pair long, and it needs to group that up into sections of 3. So right here we have our 3 codons. We have AUG, CCG, AGU. Those are what we call a codon, a group of 3. These codons on the RNA molecule correspond to a matching anticodon on the tRNA molecule. So when this tRNA molecule comes into the ribosome, its anticodon, which you can see right down here, the anticodon, is looking for the matching codon pair. So it's looking to match up with a codon on the RNA. It's going to match up regarding our base pair rules. So it's going to use those base pair rules to match. So that's apples under the tree and cars in the garage, which means that the codon that matches this anticodon, UGG, is ACC. I'm actually going to do that in a different color so you can see it a little bit better. So the matching codon for this anticodon is ACC. When the tRNA finds those 3 letters, it's going to add its amino acid that it's carrying right here to the growing amino acid chain. We have a different picture of that right here. You'll see this animated in the activity on Pivot as well. So we have our RNA strand with the letters here as the codon. Let's say it's AUG. We have our tRNA. The matching tRNA for this would be UAC, and it's going to bring with it this amino acid right here, so amino acid AA. Then the ribosome moves along in this direction down the mRNA strand. New tRNA molecules come in, and more amino acids are attached to each other to create the growing protein. But the question is, how does the tRNA, or how do we know what amino acid that tRNA molecule is carrying? How do we know what these amino acids are? There are 20 different amino acids in the human body. That's a lot of amino acids to have to memorize. You do not need to memorize all 20. Not like you have to know the base pair rules, that is. Instead, we're going to use a tool to translate our amino acids from our RNA. So just like if you were to translate from one language to another, so say you're in Spanish class and you're not sure how to do your homework, you got to translate, maybe you look on Google Translate to help you out. So you're going from English to Spanish using a translator. We're going to go from RNA to a protein also using a translator. And there are two different kinds of tools that we can use to do that. The tools that we can use to determine the amino acid sequence when given our RNA codons are the codon wheel and the codon chart. They both will get us the same answer, but there's two different ways of going about it. You can choose the one that is most comfortable for you, which can be the one that you use frequently. And I will provide both of them on the assessment for this unit. However, you should be familiar with both types because we don't know which one is going to be given to you on the biology MCAS test in the spring. It could be either one. So you should be familiar with how to use both of the charts in case you end up with the one that you are, less used to practicing. So let's look at both of them using the codon AUG. AUG is actually what we call the start codon. So every single gene sequence that you transcribed in the pivot activity started with these three letters, A, U, G. This signals the ribosome to begin the process of translation. So let's look at what the first amino acid is that's going to come into play to start off translation. Starting with the wheel, we are going to find the first letter, A, in the center of our wheel. So here we have the letter A. Then we're going to use the second letter, U, in the next part of the wheel, right here. Then we are going to use the third letter, G, in the third part of the wheel, right here. That tells us that the amino acid methionine is the one that is the start code, is the one that the start codon codes for. So this is the amino acid methionine. Let's look at the same one using the chart. So starting with the first letter A, on the left-hand side of our chart, this is where we find our first letters, we're going to find the A. This A means that any of these amino acids could be the possible answer to a codon that starts with the letter A. So let's highlight all of them. But we got the second letter. So the second letter is U. So looking at the top here, let's find the second letter. Here's the U. Any of these could be an amino acid with the second letter U, but we really only care about the ones that are also highlighted purple. So we're going to focus in on that and then find our third letter, which is G. So your third letter is all the way over here at the right. We're only concerned about the ones that are also highlighted purple. So we're going to find the letter G down here, carry ourselves across, and the one that all three colors intersect is our answer. That is our start codon methionine. So the same one that we got from the wheel. Let's do some practice now with both the wheel and the chart as we look at some other codons. CAA, the first one. So our first letter is the letter C. Our second letter is an A. And the third letter is an A. This is the amino acid glutamine. If at any point you need to pause, please feel free to do so as you complete these on your own and then pick back up and follow along with the video. If you would like to try to challenge yourself, you can pause it now, complete them on your own, and come back to press play in order to see if you got the right answer. The second one is UAC. So our first letter we're going to find in the middle is a U, and then we have an A, and then we have a C, and that is the amino acid tyrosine. Our third one, GCA, I'm going to pop over to the chart now for this one. So for GCA, let's take a look. Our first letter is a G. So we're looking at any of these. Our second letter is a C. So we're going to be down here. And our third letter is an A. So over here, follow it across. The one where all three rows intersect is right here. It's alanine. AAU. Let's start with the A. Any of these. Then we have another A for our second letter. And then we have the letter U over here. So where all three rows intersect is asparagine. Kind of looks like asparagus in my mind. Now the last one, UGA. U is over here. G is over here. And then A right here. That codes for the word stop. Stop is not an amino acid. Stop simply means to stop protein synthesis. It means that you have reached the end of the gene and the ribosome gets the stop signal to know, I'm done, the amino acid is finished, we can release the mRNA, we can release the tRNA, and we have completed the protein synthesis for that gene sequence. So there's always a start codon at the beginning and a stop codon at the end of every gene that needs to be transcribed and translated. So let's put this whole process together now as we review the 2 steps that we've learned this week. So the first step is to go from DNA to RNA, and that is the process of transcription. So we have our base pair rules of apples under the tree, cars in the garage to help us transcribe this DNA and turn it into RNA. So pause the video, try it on your own, and then come back and press play and see if you got the right answer. Hopefully you got the right answer. Let's take a look. We've got a G, U, C, C, U, A, C, U, A, C, G, A, G is the correct answer for that mRNA sequence. Now the next step is to turn this mRNA sequence into an amino acid. And that's what we just learned about. That's the process of translation. Before you go any further, take a moment and separate out your mRNA strand into the codon, the 3-letter sections. The easiest way to do that is to just draw a line every 3 letters. Keep yourself organized and so that you don't lose your place. Pause the video now, use the chart up top, and see if you can figure out what amino acids correspond to these codons. Let's see how you did. So let's take GUC, our first codon. We're going to come up here. We're going to look at our wheel. So we have G in the middle, and then a U, and then a C. So our first amino acid is valine. I'm going to write these in, I'll write them in purple. So we have valine right there. Next we have CUA. So let's do that again. Our first letter is a C, and then a U, and then an A. That's the amino acid leucine. I'm just writing the first 3 letters because they're kind of long and this just saves us some time. We know what we're talking about. So that's leucine. Next we have CUU. Let's do the chart for this one. C, U, U. Also leucine. Notice how leucine actually, if I erase all this, has quite a few amino acids. There's quite a few codons that code for the same amino acid of leucine. It appears a few times on this chart. So that's the case for a lot of them. There are duplicates, and that's okay. Doesn't mean you got it wrong. It's still going to be leucine again. That's helpful for the cell because only one letter is off still means the same amino acid. If there were some type of mutation with that letter, you could still have the same amino acid, and that's not going to cause an issue for the cell. Let's do the next one, AAC. Go back to the chart again. So A, C is asparagine. There's our asparagus-like one. I'm just going to write ASP. And then the last one, GAG. I'm going to go back to the wheel for this one. So I've got my GAG is glutamic acid, or GLU. But if you happen to use the chart for that one, you might say, wait a minute, GAU, or GAG, sorry, is on this chart something a little different. If we look at the G over here, A, U, or G, is actually glutamate. Well, glutamate and glutamic acid are the exact same thing. It's kind of like how enzymes often end in the letter ASE. Acids also end in the letters ATE, so it's just another way to write glutamic acid. They are the same thing. That is still the correct answer. So either way, it's the letters GLU for that third amino acid. So hopefully you had the right answers. If not, and you missed something, go back and rewatch the video and make sure that you practice this, because this is something that you're going to need to know, not just for this unit and for this unit test, but also for the MCAS. If you're all set, then you can go ahead and submit your notes to Google Classroom. If you did this on paper, please take pictures and submit those pictures for your classwork grade for today. Thanks for listening.